Dear student, welcome to Biospirit. Today I am giving second PUC Biology passing package. I hope it is helpful for your annual exam. So if you studied these questions means 100% you will not pass, you will score more marks. So first I will give chapter wise important questions means passing package. First one is a draw a neat label diagram of anatropous oval. So if they are asked like that question means you have to draw like that diagram. And next question is there second one, explain the transfer section of young anther. It is most important, many times they arise a question, so you have to draw like that. And third question is there, describe the structure of mature female gametophyte in angiosperm. So you have to describe the points. First one is that it is a 7-celled 8 nucleated structure, and single functional megaspore undergo 3 mitotic division to form as a 8 nucleated structure, and later it differentiated into the 7th cell. First one is that it is become egg aperture. Egg aperture means they contain one egg and 2 synergid cells, and egg cell is involved in the fertilization, and synergids it is helpful for the guiding of the entry of pollen tube into the embryos. Next is their central cell. Central cell means the two polar region polar nuclei, it will start moving towards the center and it is become as a polar nuclei, and both they fuse to form as a secondary nucleus. And next in chalazal region they have 3 cells, that is antipodal cells. It is helpful for the nutrition, it is providing the nutrition. And next question is there, describe the outbreeding device that prevent the autogamy. So in this, how the self pollination is avoiding and how the cross pollination is taking place. So many methods are there. First one, that dicogamy, that is different maturation time, means the anther is maturing one time, and so ovary means gynoecium it is maturing at another time. Protandry, in that sub type is there. So anther is maturing in another time means then we are calling as a protandry. If the gynoecium it is maturing at the different time means then we are calling as a protogyny. Andry means androecium, gyne means gynoecium, proto means it is maturation is there, which one is maturing first, that is. And next hercogamy is there. Hercogamy means physical barrier between the anther and stigma, means they have a different in length. Next, self incompatibility. Self incompatibility means the stigma have one type of protein, and pollen grain also they contain one type of protein. They checking it is same type of protein is there means they will think it is our plant pollen grain is there, so they rejecting. And next it is unisexuality is there, means in some plant the male reproductive structure present in one flower and female reproductive structure is present in another flower, that is we are calling as a unisexuality. Next, heterostyly. Heterostyly means the style length is there or different length of the stamen or style. Next question is there fifth one, briefly explain the structure of pollen grains. So how to explain the structure of pollen grains. Outermost layer we are calling as a exine, it is made by sporopollenin, and innermost continuous layer is called innermost layer is called intine, and it is made by cellulose and pectin. And outermost that exine covering is there. In certain reason exine is not covered, that area uncovering of exine we are calling as a germ pore. It is helpful for the developing of a pollen tube. Inside they have 2 cell, one vegetative cell, another one generative cell. Vegetative cell helpful for the pollen tube formation, generative cell it is helpful for the gamete formation. Next question is there sixth one, draw a neat label diagram of longitudinal section of the typical flower. So you have to draw like that. And next question is there, draw a neat label diagram of the monocot embryo and dicot embryo. These are the monocot embryo and dicot embryo structure is there. Question, eighth question, list out the functions of tapetum. Tapetum it is providing nutrient to the sporogenous tissue, and second one is that it secretes sporopollenin and it is used for the pollen wall formation, that is exine, and it secretes the Kalisch enzyme. It is helpful for the degrading the callose wall surrounding the microspore tetrad, and it secrete compatibility protein, that is it is helpful for the self incompatibility. And next it is produce substance such as pollen kit. Next, ninth question is define the following terms: Clistogamous, flower, and parthenogenesis is a gytonogamy and genogamy. These are the definitions. Next question, write any 4 characteristics of the flower pollination by winds. So first one is that these are the flowers typically small, lack and bright colors, do not produce nectars, they're not producing any nectars, and produce large quantities of a dry and light and dry pollen grains. The stamens are often long and it releases pollen into air. Many wind pollinated species have a unisexual flowers, and the stigma are typically large and feathery. Next question is here, how does pollination achieved in Valsinaria and seagrasses? So these are the points, you refer it. Chapter number 2, human reproduction is there. From this chapter, first question is there, draw a neat label diagram of male reproductive system. So it is male reproductive system, you have to practice it. And next question is there, draw a neat label diagram of the female reproductive system. Question is there, third question, draw a neat label diagram of the sperm. It is sperm diagram is there, you have to practice. And next question is there, draw a neat label diagram of the mammary gland. It is mammary gland, you have to mention all the labelings. And next, mention the accessory duct of the female and male reproductive system. So female reproductive accessory ducts are there, fallopian tube, cervix and vagina, and male accessory ducts are there, retestis, vasa efferentia, epididymis, vasa deferens, ejaculatory duct. Question is there, sixth question, write the schematic representation of the spermatogenesis, and second one is there, oogenesis. It is spermatogenesis schematic representation is there, and it is oogenesis schematic representation. And next question is there, list the hormones produced by the placenta during the pregnancy. So first it is producing human chorionic gonadotropin and human placental lactogen, estrogen, progesterone and relaxin. These hormones are released by the placenta during the pregnancy. Question is the eighth one, what is pasteurization? Which hormones are involved in the induction of the pasteurization? Pasteurization, it is the process of giving birth to the baby. So it is delivery process is there. And second point, many hormones are including oxytocin, estrogen, progesterone and relaxin, and placenta which induce a mild uterine contraction called fetal ejection reflex. It is most important is there, many times they arise the question on fetal ejection reflex. Next point, it trigger the maternal pituitary to release the oxytocin hormone. Next chapter is there, fourth one, that is reproductive health. From this chapter, that is fixed questions are there, mention any 4 sexual transmitted disease, that is gonorrhea, syphilis, AIDS, genital herpes and genital warts is there. And next, second question, what is infertility? How infertility treated by assisted reproductive technology, that is GIFT and ICSI. So it is most important is there, many times they arise this question. So you have to write these points. Next, third question is here, what is medical termination of the pregnancy? Mention the safe periods. So medical termination of pregnancy means MTP refers to the intentional termination of the pregnancy by using medical or surgical methods. So what are the reasons they need for the termination of pregnancy means? That is, first one is to prevent the unwanted pregnancy. So, and second one is in case of rape, the female is incident caused by. Next, when the pregnancy proposes a risk to the mother physical or mental health. If that pregnant woman is not suffering by mental health, means in that case they can remove the fetus. And next, so if they've caused by genetical disorder means they can remove the fetus. And if that baby is severe abnormality means that is hole in heart and some organs are poorly developed, so like that cases are there means they can remove the fetus. So this MTP they can refer, that is first and 12th week, and also they can refer 20 and 24th week, but it is most risky situation is a this case, that is after both the 20 or 24th week, if the female is ready to remove the fetus means, so maybe the female they can suffer by a critical issues, maybe the female can die. And next question is there, what is contraception? Explain any 4 contraceptive methods. So contraception means a method which allows the sexual intercourse but it prevents the pregnancy, that is we are calling as a contraceptive. And they can follow say IUCD method, intrauterine contraceptive device, or intra contraceptive device, or they can follow by pills, can follows by a surgical methods, or surgical means first vasectomy is there, second one tubectomy. Vasectomy means removal of, that is vasa difference. Tubectomy means the surgery takes place at the fallopian tube. And some natural methods also they can follow see for contraceptive purpose. And next chapter is a 4th chapter, principles of inheritance and variation. From this chapter, first question is there, what is incomplete dominance? Explain with suitable example. So incomplete dominance definition is there, the dominant alleles are partially expressed, often resulting in an intermediate phenotype. Means the 2 dominant allele is there, one dominant allele they express partially in the presence of another dominant allele, so they showing the different type of the character. So here you take example, that is snapdragon is there example. So red flower cross with the white flower, they giving pink flower because the dominant allele they express partially. So it is phenotypic crossing is there. Next, second question, state the law of independent assortment and write the schematic representation of dihybrid. So first, this law of independent assortment statement is that the different genes are sorted into the reproductive cells independently of each other. Means here you take example, that is round and green seed is there, 2 character, one that is shape we taken, another one color of the seed we taken. So both trait alleles are individually they entering in each gamete. You can check, round 1 allele is entering and green 1 allele is entering. So this is we are calling as a law of independent assortment. So it is schematic representation. Third question is there, explain the Mendel experiment to describe the inheritance of one gene with reference to the height of the pea plant. So that is, so you have to practice this crossing. And next is there, mention the causes and effect of the followings: phenylketonuria, Turner syndrome, Down syndrome, and Klinefelter syndrome, and sickle cell anemia. These are the points, you read it. Students, once again I am repeating, if you studied or if you make perfect these questions means 100% you will score more marks. Next, 5th question is that describe haplodiplomatic sex determination system in honeybees. So here 2 type is there honeybees, one that is female, another one male. Female one we are calling as a queen, which is able for the reproduction. So male one we are calling as a drone. Female they have 32 chromosomes, drone they have 16 chromosomes. Means the female they producing 4 gamete because they have a diploid chromosomes, double set of chromosome is there. Male they have only half set of chromosome, so that's why they're producing only 2 male gametes. So one male gamete fuse with one female gamete and they become as a 2n, that is female. Another female gamete they fuse with the another male gamete, they become as another female honeybees. And remaining 2 female eggs are, it is remaining because they don't have any male gametes, only 2 are there, they fused with 2 female gametes. So the 2 female gametes are left over, they waiting for the male gamete but they not get, means it is start develop, become a male, that is drone. If the 2 females are formed, the females they get royal food, means they become as a queen, and if they not get royal food means they will become as a worker bee. Next, fifth chapter is a molecular basis of inheritance. From this chapter, first question, let's see silent features of Human Genome Project. So first point is that the human genome contain 3164.7 million base pairs. Second point, the average gene consists of 3000 bases, and the total number of genes is estimated at the 30,000. Almost all 99.9% nucleotide base pairs are exactly same in all peoples. Less than 2% of the genome code for the proteins, and chromosome 1 they have most gene, that is 2968. So that's why it is largest chromosome is a smallest, that is Y chromosome, because they have very least genes, that is 231, and repetitive sequence make up the our very large portion of the human genome. Next, second question is that describe Frederick Griffith experiment to show the transformation in bacteria. So it is one experiment, it is conducted by the Griffith in 1928. So explanation is here. If he taken newborn mice, and plus he taken two type of the bacteria, that is Streptococcus bacteria. One is a rough smooth Streptococcus strain, and another one is a rough Streptococcus strain. So in one mice they injected the smooth Streptococcus bacteria, that is S strain they injected, and mice is dead. And next, if they he taken another one mice and he injected the R strain, and mice is living. And he taken third one mice and he injected the heat killed S strain, and mice is living. And next, in fourth mice he taken and he injected heat killed S strain plus R strain, and mice is dead. By this experiment he concluded that is heat killed S strain, if it is mixed with the R strain, means the transformation of genetic material is takes place, so that's why the mice is dead. Next, third question, describe Lac-Operon concept with labeled sketch. So in E. coli, how the metabolism is takes place, so that is explaining in Lac-Operon concept. So we'll discuss. So first you check these points, and here is a switch off. So in E. coli they have a DNA stand. In DNA stand they have some controlling genes, that is promoter, inducer, promoter, and operator, and structural gene. So it is DNA stand is there. On DNA stand the RNA polymerase is start moving once it is reach promoter, and next it start moving to the inducer region. In inducer they start transcribing one small mRNA fragment, and mRNA fragment it start synthesizing the protein, that is we are calling as a repressor protein. So this repressor protein it will wait for the sometimes, because in E. coli they have a lactose, means the lactose will be bind to the repressor protein and it will start working, and that is we are considering as a switch off. But here I am explaining switch on, but here I am explaining switch off, because here there is no lactose in E. coli, so that's why the lactose is not bind to the repressor protein, and after sometimes the repressor protein will be moving towards the operator region and it will block the operator region, and RNA polymerase will reach to the near the operator. Once it is got the repressor protein, it is blocked the operator, means it will exit out, and after exiting there is no mRNA transcription, there is no synthesis of any enzymes, and there is no breakdown of the lactose. So this is we are calling as a switch off. Other one is there, that is switch on in the presence of inducer. So same is there in E. coli, they have a DNA fragment. So RNA polymerase will be start moving towards the DNA fragment. Once it is reach promoter and it will pass towards the inducer region. Inducer they transcribe repressor mRNA, and this repressor mRNA they start synthesizing one protein, that is we are calling as a repressor protein. And in E. coli they have a lactose, it will bind to the repressor and it start become as a inactive, and after that here there is no blockage because it is locked by the repressor protein, it is locked by, that is lactose. And RNA polymerase, it start moving towards the structural gene, and they're reading the structural gene, that is ZYA, and they're transcribing the mRNA. So this mRNA, it is synthesized by 3 genes, so that's why they're producing 3 enzymes. One is their beta galactosidase, another one permease, another one translation transacetylase. So this synthesis will be takes place, and the beta galactosidase, it is activated by translation transacetylase, and the permease, it is controlling the level of the lactose, and the beta galactosidase, after activation, it start breaking the lactose. Next one is there, fourth question, write the any 5 silent features of genetic code. So first one is there, the codon is a triplet, and 61 codons code for the amino acid. So it is triplet codon is there, and they have a 61 codons of a amino acid. Some amino acid are coded by the more than one codon, so that is we are calling as a, hence a codon is degenerate. And next one is that the codon is read in mRNA in continuous fashion. And next, AUC is initiation codon, it's code for the methionine. And stop codon or nonsense codons are there, UAA, UAZ, UZA. And next question, fifth one, explain the process of transcription in eukaryotic. So these are the points, you can refer it. Next question, sixth one, mention the steps involved in DNA fingerprint. So in DNA fingerprint, first step is a sample collection. So we are collecting sample in the form of, that is blood, hair, semen or skin cells. And DNA extraction, so that collected sample we are treating by enzymes, and that after treating we are extracting the DNA. Next, DNA digestion by the restrictive enzymes, that isolated or separated DNA we are cutting in different length by using the restriction enzymes. And next, after that cutting, the samples we are running in gel, that is we are calling as a gel electrophoresis. Means in this process we are separating the DNA fragment based on their length, and afterwards we are following the Southern blotting. In that Southern blotting, that we are transforming, that is DNA fragment from gel to the nylon membrane or intracellular membrane, and after that transforming into nylon and nitrocellular membrane, we are treating by probes, radioactive probes. Means by this process we can detect the specific DNA sequence. And after that treating the probes, we are checking under the autoradiograph. And next, comparison and analysis. Once we checked in autoradiograph, we can check, that is matching band form takes place between the that who did crime and who is already is crime dead person is there. So we are checking dead person and crime person. So if it is matching, means we can say that particular person only they did crime. And next question is there, write the schematic representation of the central dogma. So here DNA, they're duplicating DNA, that is process we are calling as a DNA replication, means duplicating of the DNA by using DNA. Next, the DNA we can use for the synthesizing of RNA, that is we are calling as a transcription, and RNA we can use for the synthesizing of a DNA, that is we are calling as a, that process is called as reverse transcription. RNA, it is starts synthesizing the protein by the help of, that is translation. Next, eighth question is that describe the process of DNA replication with the help of diagram. So it is first point is there. So the DNA replication means duplicating of the DNA is called DNA replication. So here 3 main steps are there: initiation, elongation, and next last one, that is joining is there, means termination. So first what will happen, the helicase will be enter in the DNA strand, and it starts separating the DNA fragment DNA fragment. Helicase, it will break the hydrogen bond. Due to the breaking of hydrogen bond, it will start separating the DNA strand, so it will become as a frog like structure. And next, after the separated DNA strands, it is binded by the single strand binding protein. This protein, it is preventing the rehelic or rejoining. Next, topoisomerase is there. In front of that is open front region, they have supercoiled DNA, so that is removed by supercoiled DNA is prevented or removed by the topoisomerase. And next, after this process, elongation, the primer will be bind to the open region, and it start here taking the help of DNA polymerase 3 and extending the new DNA strand. So here two type of new DNA strand will be formed, one continuous, another one of short many fragments, that is we are calling as a Okazaki fragments. And this short one Okazaki fragments is joined by DNA ligase. So these points you have to write. And next question is describe the process of translation mRNA, translation of mRNA. These are the points are there, you can read it. These are the continuous points are there for translation. And next, evolution chapter. From this chapter, first question is draw a neat label diagram of the Miller's experiment. It is most important. Many times there is a question on this topic, you have to practice this diagram. And next question is differentiate between the homologous and analogous organ. Homologous means origin is there, that is same. Ana means origin is different. In homologous, origin is there same, but the function is different. And in analogous, origin is there dissimilar or different, but function they will same. Example, you take arm of the human and leg of the dog, and here you take analogous, that is wings of the bats, butterfly and insect. Question is third one, write a short note on the adaptive radiation. These are the points are there, you can read it. Next, it is most important is fourth one, that is what are the factors affecting on the Hardy-Weinberg principles, that is mutation, recombination, genetic drift, gene migration and natural selections. These are the effect on the Hardy-Weinberg principle. And next, seventh one is human health and disease, that is draw a neat label diagram of the structure of antibody. It is most important you practice this diagram. Next question, write the diagrammatic representation of the life cycle of a HIV or retrovirus. It is schematic representation of retrovirus is it. You practice. For annual exam, the diagrams they are arising on 13 marks. Total 30 marks questions will be related to the diagrams only. You have to practice the diagrams. And next, third question is name the disease caused by following organisms: rhinovirus, epidermophyton and Salmonella typhi. Next, Uginaria malai and Plasmodium vivax. These are the disease caused. Rhinovirus, it causes a common cold, and epidermophyton, that is ringworm is and Salmonella typhi, it will cause a typhoid. And Uginaria, it is called the elephanthus. Elephanthus in Kannada we can call that is Anekalu roga. And in Plasmodium vivax, it will cause malaria. And Streptococcus, it is I mentioned extra, Streptococcus pneumoniae, it causes pneumonia disease. Next question is differentiate between the active immunity and passive immunity. These are the difference are there, active and passive, you refer. And next question, briefly explain any three prevention and control measures of the drugs or alcohol abuse. It is most important, many times they arise this question, so these are the points, you read it. And next chapter, microbes in human welfare. From this chapter, the most important diagram is draw a neat label diagram of the typical biogas. And next, most important diagrams, draw a neat label diagram of the PBR 322. And next one is draw a neat label diagram of the stirrer tank bioreactor. It is also most important. So dear students, these are the important questions are there for annual exam. So if you study these questions means you will pass and you will score high marks. So if you like our videos, like and share, subscribe. Thank you for watching.